They were there in this place for 6 months. So if you look at this place, I have just put some simple data over here. Microgravity. Can you avoid microgravity? You cannot. So we are doing research in our laboratory from last 8 to 10 years and published several papers to show you that how microgravity can induce DNA damage and it can cause cancer. So far people are ignorant about the microgravity. Everyone is talking about the radiation. But microgravity is also having very strong potential to induce DNA damage. So this is just one data to show you. This is called as cometation. Look at the left hand side. You are seeing that red color is moving on the backward side. Those are the small fragments of DNA. If you do cometation, you can see how at what level SMG simulated microgravity, what level of DNA damage has been caused. The right hand side, 2D culture versus spheroid. Microgravity has very strong potential to develop spheroids. We have done several experiments in the spheroids to show what mechanism and how does it changes the characteristic of the cell. So this is just one slide to show you the effect of the microgravity. Obviously, the life sciences 2 months back. And here we have explored the DNA damage and DNA repair, how it is getting affected from the microgravity. So, we will not go into the detail. If you are interested, you can read the paper. So, in the next 10 minutes, I will just discuss some of the experiments, and this will be the last part of the presentation on the prostate cancer. So, prostate cancer, in the normal epithelial cells, mutation is happening, getting converted into the adenocarcinoma, and that is how the cancer is diagnosed. And at this time, it is usually androgen receptor positive or androgen positive. So whatever drugs are given, they are targeted to either androgen or androgen receptor. What happens after some time? 1 to 3 years, the tumor is again coming back. This is called as the relapse of the tumor, and it is regrowth. So when relapse is happening, that time the tumor is very aggressive. It is not going to respond to the earlier drugs. Most of the drugs are not going to work, and you have the options for radiotherapy or chemotherapy. So when you go for radio or chemo, again it will become resistant to the radiotherapy. It will be radio resistant or resistant to the chemotherapy, chemo resistant. So here we explore the mechanism of radio resistance. That is a very novel mechanism. Next. So for radiotherapy, it is very common for the cancer patients. And almost more than 50% cancer patients, they go for radiotherapy for the solid tumor. That is already there. Data for the treatment. What problems we face? The development of resistance and the toxicity. So these two are the major problems of radiotherapy, also for the chemotherapy. So how to overcome these limitations? You have to have a strategy where you counter the resistance and also reduce the toxicity. So we took a very simple compound. This is silimarin. This is a phytochemical flavonoid. If you look at its 3D structure, you find that this molecule has both hydrophilic as well as hydrophobic structure, ranging from blue to brown. So this tells that this molecule can perform a range of interaction, which can be from hydrophilic to hydrophobic. And this molecule can enter into the cell as well. So we did a very simple experiment. These are called as athymic mice. Athymic does not mean that they don't have thymus, but they are immunocompromised. What is the advantage of this mouse model? You can grow human tumor on mouse. So graft will not be rejected. So this D145 is human prostate cancer cell line. You inject them subcutaneously, tumor is growing. Left hand side versus right hand side. Right hand side, you will find the treated group where you have the small size of the tumor. So So first observation that whatever drug you are using, the drug is inhibiting the growth of the tumor. Now your idea is to find out what could be the molecular target. This EGF receptor, most of you might be knowing, epidermal growth factor receptor. And this signaling is very common for growth and survival of the cancer cells. So we did a very simple experiment over here. You see 9L cell line is a red glioma cell line which does not express EGF receptor. So we made a construct and expressed human EGF receptor in this mouse red 9L cell line. So in the first two, you can see the dose of the drug 2, 5, 7.5 and 10 micromolar silibin plastic. In the first two rows, you didn't see any difference. Those purple dots are the colony of the cells which are growing. When you look at this third one, 9L EGFR. So here EGFR is expressed. Now you see the effect of the drug, which is dose dependent. You see that those cells colonies are disappearing when you increase the dose of the drug. This A431 is a skin cancer cell line known for high expression of EGF receptor. So here also we find almost similar trend. So this tells that when EGFR is expressed in the cell, your drug is working. If it is not expressed, your drug is not working. So EGFR could be a potential target of the drug. So this is one way of how you find this target of the drug. And why it is interesting? Because EGFR is expressed in almost more than 40-50% of cancers. So, since you are talking about the radiation, if you look at just the bar diagram, that right corner, just focus on that bar diagram. This is the data related to reactive oxygen species. First one is control in D145. Next one is silibin, and then you have radiation, and then combination. You have 4 bars. The black bar is the combination of radiation and drug, where you see high level of RS formation. Drug itself and radiation, they are also causing RS formation. So left hand side D145 is prostate cancer cell. Right hand side HEK293. This is a non-cancerous cell line, non-neoplastic. If you look at that, you will find the difference in the bar patterns. What you have left hand side is a cancer cell versus in the normal cell. What is happening in the normal cell? If you look at the third bar is for the radiation treatment. The bar height is same in both cancer cell and the normal cell. But your drug is differentiating between the normal and cancer cell. Drug is not causing high level of RS in the normal cell. Even when you are combining, the last one black bar, the drug is inhibiting the RS produced by radiation in the normal cell. So this is a very important data in terms of selectivity of your drug for the cancer cell versus normal cell. So this was a very important observation. Then since we were interested in defining the target of the drug, this is EGFR, how it gets activated. This is the classical mechanism, left hand side, ligand-induced activation of the receptor. You might have read in your cell signaling class. Right hand side, what I am showing you is stress-induced activation. This is ligand-independent activation of the receptor. That is also happening. And there may be several situations like UV, ionizing radiation, some of those RS or oxidants. They don't bind to the extracellular domain of the receptor, but they can activate the receptor. And you will have the same response. So, left hand side, that is the conventional part of the EGFR signaling. Right hand side is very unique part of the EGFR signaling. What is unique here is this EGF receptor is entering into the nucleus. Look at that structure, that double structure which is there on the membrane, 2 bars. They are entering into the nucleus on right hand side. That is why it has written NEGFR, that is nuclear EGFR receptor. So EGFR receptor is 170 kilodalton molecule. It is supposed to work on the